I would like to introduce Paul French to our LIBREhub seminar series. It's a special pleasure to have Paul here because he's actually one of the senior people in the open hardware community. So he's already been building and disseminating microscopes long before the sort of recent exponential growth of the topic and the community. He's done a lot of very impactful work, and it's really fantastic to hear from him. So he's a professor at the Imperial College London, a beautiful and a very good university in London. And his institute is also a satellite laboratory of the Francis Crick Institute, if I remember that correctly, which is a really, really cool, recent, high-profile research institute they set up in London to bring together top researchers. He's done his bachelor actually and PhD in laser optics also at Imperial College, I think. And you've worked at a number of different institutions also in the US in between, including the Bell Laboratories, if I understand correctly. That also sounds really, really interesting. We haven't actually talked about that experience yet. I think that must have been a really interesting place to work as well. But then coming back home to drive his multidisciplinary research on a number of optical aspects, going into some dye research, I think, some solid state research, but also especially what brings him here today, modular microscopy that can be applied to biology and its open source dissemination. So thank you very much for being willing to present in our seminar series, and we're very excited to hear from you, Paul.
Thank you very much, Tobias, for this invitation. I'm going to talk today about our work on what we call OpenScopes, a platform for open modular microscopes where we're aiming to widen access to microscopy and high content analysis capabilities. And I'm not sure I'd agree that Imperial is a beautiful university. I think it's not particularly, it's a fairly ugly set of buildings, but in a beautiful part of London. It's not the oldest university in the UK. We've been around since 1907, but we've been doing optics more or less continuously since 1917, I should say, not 2017. And yeah, so it's one of the older optics sections in the world. And so, as Tobias mentioned, we have laboratories mostly based in the physics department at Imperial College, but also we have a satellite laboratory at the Francis Crick Institute. And in these laboratories, we work on a wide range of different microscope capabilities, ranging from super resolution, recently correlative light electron microscopy. My colleague Chris Dunsby has invented and pioneered the technique of oblique plane microscopy, and we use that for both well, he uses that in a wide range of applications, including high content analysis. We've also worked for many years on fluorescence lifetime imaging and had a particular push there to implement it in multiwell plates for high throughput, high content analysis. And recently we've combined that with quantitative phase imaging, with multiphoton microscopy. And we've also worked on preclinical imaging, doing fluorescence lifetime imaging, FLIM and FRET, in a mouse cancer model. We've built an optical projection tomography system for imaging both fixed and live samples. And we've done a little bit of clinical research using the Derm-Inspector clinical multiphoton microscope pioneered by Karsten Koenig. And so the group in the physics department is led by myself, Chris Dunsby, James McGinty, and Mark Neal. And I'd particularly like to acknowledge the many contributions over the decades of our postdoctoral researchers, research fellows, Yuri Alexandrov, Edwin Garcia, Sunil Kumar, Jonathan Lightley, and Hugh Sparks, amongst many others. These are the people who've particularly contributed to the more recent development of the openFrame and OpenScopes microscopes. And so over time, the goal of our lab has been to apply techniques in photonics, biophotonics to imaging and microscopy, and particularly fluorescence imaging, where the goal has been not just to image with intensity. So essentially, fluorescence imaging with intensity readouts tells you what is the spatial distribution of your fluorophore, and therefore the target, the proteins that you've labeled. We've also combined intensity imaging with spectroscopic imaging. Of course, you do that first with wavelength. Different colors can label different species, and you can look at co-localization. If you want to look at interaction, then you can use techniques like FRET, and we would say FRET is best done most quantitatively using fluorescence lifetime imaging. And also you can gain information about molecular orientation and dephasing if you resolve your fluorescent signal with respect to polarization, and you can look at dynamics if you make time-resolved instruments. And so we've been doing this for a long time, and we've built a lot of different microscopes. And being physicists rather than biologists, we're not slowed down by a biological question. And after we've built our microscope and demonstrated hopefully some novel capability, we typically take it to pieces and build another one. And so, as Tobias said, we've been building our own microscopes, writing our own software for a long time. And basically, now I'm much nearer the end of my career than the beginning. If I think about what's important for the future going forward, at least for what I'm interested in, I think it comes down to capability, these different modalities that we can build, spectroscopic imaging, applying microscopy to 2D, 3D samples, super resolution, quantitative phase imaging, but also to throughput and to accessibility. Throughput is really important for statistical robustness. If you're trying to study cell biology, typically your samples are heterogeneous. You get different responses, and you need to look at many samples to average over the biological variability. And accessibility means making these capabilities more widely available. For a long time, particularly in the physics community, it seems like it's a race to do things first, and there's a certain amount of kudos in being able to do something that no one else can do. But really, in the wider scientific community, what is the point if no one else can do it? And so we want to make our instrumentation more accessible, and this means generally it has to be lower cost. If we're thinking about trying to reach low and middle income countries, low resource settings, it needs to be locally sustainable because communities there often can't afford the standard service engineer based model. Typically it wants to be modular, so it can be upgraded. It wants to be open source and use generic components. And so at the heart of our open source, open microscopy program is the openFrame-based microscope, and this is a schematic of it here. And so I wanted to say something about throughput. When we're doing cell biology, it's really important to not just look at a few cells in a field of view, which typically might have hundreds or thousands of cells in, and just look at the ones that show the result that you want. It's much better if you want to have robust, reproducible biology to do automated imaging of thousands of cells in hundreds of fields of view, and to have an automated, unbiased data analysis. So this is the essence of high content analysis, for example, applied to cell-based assays. And of course, it's a standard in drug discovery where they would have automated cell culture, liquid handling, automated cell resolve readouts. And essentially, you're moving from the microscope slide to the multiwell plate. And what's really important here is you can do multiple experiments under the same conditions, samples prepared in a consistent way. You can have repeats of experiments so that you make sure you're not looking at something that does the wrong thing by chance. And you can have controls so that you can make sure that you're measuring a real effect and not an artifact of something that you did control for. And of course, this can be applied to live or dead fixed cells. There are many kinds of readouts. You can look at morphology using brightfield imaging or quantitative phase imaging. You can look at fluorescence. You can look at gene expression, translocation, biomolecular interactions, and so on. And so what I'm going to talk about in this talk today is I'm going to kind of continue the motivation and how we got to open source microscopy, starting with our work on FLIM and FRET. And then I'm going to talk about open single molecule light microscopy, which was the kind of stimulus to make us come up with OpenScopes. I'll talk about easySTORM, which is our implementation for low cost large field of view, SMLM, an application in histopathology, I'll briefly mention the autofocus, which is a critical capability for time-lapse and multiwell plate imaging. And I'll mention some recent work done with Lucy Collington at the Crick, where we're looking at super resolved volumetric correlative light electron microscopy. I'll then talk about sorry, I have to move my dialog window. I'll then briefly mention another advanced application of open microscopes, what we call the M3M, multiphoton, multi-beam, multi-well plate microscopy we're applying to 3D cancer models. And then I'll switch gear a bit and talk about the work we're doing to disseminate OpenScopes and open frame instruments, low-cost modular microscopes, our OpenScopes Africa project. And in the last third of my talk, I'll talk about our new technique for quantitative phase imaging, which is very simple to implement, which we use for single cell resolved imaging, tracking, segmentation. And if I have time, I'll talk about PulseSpec, a potentially single shot hyperspectral imaging technique using spectral modulation vectors. So we worked for decades since the 90s on fluorescence lifetime imaging technology, and my PhD was in ultrafast dye lasers, and I then went on to build ultrafast solid state lasers. So I've always been biased towards the time domain, and we've built fluorescence lifetime imaging systems based on time correlated single photon counting and wide field time gated imaging. And as we did this, coming back to the point about we used to build pretty sophisticated instruments that no one else had access to, and being feeling driven to make it to have our instruments used by other people, we developed an open source system. So we developed an open source image data acquisition system, initially using LabVIEW and then progressing to Micromanager. And this was published in the Journal of Visualized Experiments. And for a long time, building particularly on the work of Sean Warren in our group, we developed a fluorescence lifetime analysis software capable of doing global analysis, which is extremely enabling. I'm not going to have time to talk about fluorescence lifetime imaging now, but this was our first open source microscope. And indeed, if you go to our website, you'll be able to find these resources. So OpenScopes really kicked off when we were asked to change gear ourselves and to work not on FLIM, but on super resolved microscopy. This was a collaboration with the Faculty of Medicine, and this is an example of single molecule localized imaging using STORM, where the Streptococcus pneumonia bacteria inside THP1 cells, which model macrophage, are labeled, and our colleagues in the National Heart and Lung Institute were interested in how bacteria in the process of phagocytosis. And here you can see that if you're doing super resolved imaging, you have a much better image and quantification of the bacteria being phagocytosed by the THP1 cells. So these images were not taken on a commercial microscope. They were taken on a home-built super resolved microscope using a technique we called easySTORM. And easySTORM is effectively an implementation of DSTORM, which was developed by Mokazawa's group, Heilemann et al. In 2008, and also is very reminiscent of ground state depletion with imaging return, a technique developed by Stefan Hell's lab in the paper Folling by al at the same time. And essentially what these amount to is labeling samples with dyes or even sometimes fluorescent proteins that you already use, putting them in a convenient medium that makes the emission spontaneously blink when you pump them with the lasers that you normally use to excite those fluorophores. And so this makes super resolution easy. And at the time, if you wanted to buy a super resolved microscope, it cost the best part of $500,000. You would typically start with a turf microscope, add the lasers, and so on. And for example, in the red, where DSTORM tends to work best, using Alexa 647, they were offering solid state frequency double lasers, excitation lasers, which were extremely expensive. And I wondered, could we do better than that? And I found that for £30, I could buy a 700 milliwatt laser. The catch was that it wasn't single mode, it was multi-mode, which meant that we couldn't couple it into the single mode fibers usually found in turf microscopes. So then we thought, what about coupling it into a multi-mode fiber? And of course that works. And if you put it in a multi-mode fiber and you have a multi-mode laser, because you have many modes, you can vibrate the fiber, and this gives you a uniform excitation over a very wide field of view. And it turns out that you can do this You can build your own diode driver, or there's a company we use called LaserTAC in Germany, which makes very nice beam conditioned multimode lasers. And typically for a few €100, you can access over a watt of excitation power. And so this is suitable for most of the dies used for SML for storm-based techniques. And you can still do turf. It turns out as long as you confine your excitation to this annulus in the outside of the back aperture of the microscope objective pupil, you can do turf. And so this is an example of fluorescence turf of an immune synapse. This is on a cover slip, and this is turf storm. And this movie, if it plays, is showing us basically imaging some cells labeled with Alexa 647 on a cover slip, and we are scanning the cover slip such that after a while, with single mode excitation, with multi-mode excitation, you lose the fluorescence. This is because you're outside the turf excitation zone, and you can see that up to 100 microns fiber turf is quite practical. And so this is our easySTORM microscope. We have low-cost lasers. We simply have a steering mirror that moves the beam either from the center for epifluorescence, the edge for turf, or in the middle for high-low excitation. And when we started doing this microscopy, we came up with 120 micron field of view from the outset. And we puzzled for a long time why it took us so long to do data analysis using the standard and indeed some of the very fast SMLM and that's software analysis packages. And we realized it was because our field of view of typically 125 microns squared was much larger than the typical 25 micron squared field of view often found in commercial microscopes. And so in order to speed up our data analysis, Ian Munro in our lab piggybacked on bioformats and developed an HPC parallelized processing pipeline where we take our stack of frames for single molecule localization microscopy and send each one to a different node on an HPC, and then we can do it much faster. And so this made it tractable, and we didn't need to buy very expensive workstations. And we then decided that if this was easy for us to do, could we do it somewhere else? And in a collaboration with Basanta Barua's lab at the Institute of Indian Technology in Guwahati, we built an upgrade of £5,000 upgrade of a standard epifluorescence microscope to STORM. And this is a picture of it in the lab in Guwahati. And these are our laser banks using the diodes I talked about earlier. And they are now have an independent research program where they're investigating ways to improve super resolution microscopy. And so this gave us the thought that if we can export, if you like, or transfer this capability to one lab, we can do it to many. And so we set up OpenScopes.com, which is a website where we tried to collect together some of our open source tools. And we thought, if we can make super resolution microscopy cheap, what could we do about applications? And so we wondered about how we could apply this medically. And for medical applications, why would you need super resolution? And the thought I had was, well, is there a medical application where people currently use electron microscopy where they need resolution below what you can get from standard diffraction limited microscopy? And my colleague Candide Rufos, who is a renal pathologist, told us that electron microscopy is used in the histopathology of kidney disease, of native kidney biopsies, where it adds value in at least 50% and is essential for 20% of diagnosis cases. And basically, this is a standard diagnostic technique in the UK, but not in so many countries. And even in the UK, the number of centres is closing because of the high cost, not only of the electron microscopes, but also of the staff to run them. And so we thought, could we replace electron microscopy of kidney biopsies with super resolved microscopy? Could we make it a simple upgrade to an existing immunofluorescence microscope? And then it could probably be operated by existing pathology staff. And so this tech could be much more widely accessible. And so we set ourselves the task of using easySTORM, of only using standard National Health Service approved antibodies. We didn't want to use any special research protocol, techniques and protocols. And we developed protocols that work with standard formalin fixed, paraffin embedded or frozen histological sections. And needing a snappy name, we called this HistoSTORM. And I should say we were not the first people to apply STORM to pathology. This was also done in the group in Pittsburgh, for example. And so this slide shows some examples. The top row are green and red channel of an immunofluorescent microscope and the overlay image. The green is labeling laminin. The red is labeling immunoglobulin G deposits in the tissue section. The middle row shows an electron microscopy section, image of a parallel section, and the fluorescence image from here blown up to here. And here in the central column, you can see the STORM image. And then if we blow up this region again, you get the bottom left image where you can see it's totally blurred, but you can still see clear structure in the STORM image. And again, here's a parallel electron microscopy section blown up. And so what we can see is if you don't have electron microscopy, it seems clear that the electron microscopy, that the STORM could add information which is not available in the conventional diffraction limited immunofluorescence microscopy. And one of the diagnostic criteria in renal pathology is the thickness of the glomerular basement membrane. And here we have laminin labeled again with bifluor 647 in an FFPE section. And if we look in a standard immunofluorescence microscope, this comes out at 657 nanometers, whereas the parallel section in the electron microscope comes out at 281 nanometers. Using STORM, we can measure in a different section 212 nanometers, much closer to the underlying reality, or at least that indicated by electron microscopy. And so we think this is an example where a low-cost open source approach to microscopy could actually make it into the clinic, and that would be a long-term dream. When you're doing STORM, you often have to acquire many hundreds, even thousands of frames, and so it's always a problem if you have drift of the microscope. And so to address this, and also to address slide scanning and multi-well plate imaging, we wanted to come up with an autofocus. And so we designed an autofocus module where we take a laser diode and we focus it onto the back underside of the cover slip, and then we image it back and separate it from the fluorescence with a beam splitter and focus it onto an autofocus camera. So the idea is that when your sample is in focus, you see a spot on the camera, and as your sample drifts out of focus, this spot becomes bigger, and that would tell you how far you were from the focal position, and then you can apply a correction to some translation on the objective lens or the sample. And so one twist we added to this was that we incorporated a slit so that instead of focusing to a point on the sample, we focus to a circle, we focus to an ellipse. And that means the confocal parameter is short in one direction and larger and more slowly diverging in the orthogonal direction. So this means that we can kind of optimize for range and precision at the same time. And this shows a movie of the autofocus camera image as the sample is translated through focus. And you'll see it's not exactly elliptical, but it has a different scale in the vertical and horizontal axis. So we published that, and it works, and it ran for months, and it was reasonably stable. We used a neural network to read out the degree of defocus from these images. That turned out not to be our smartest decision because there's a lot of time to train the data, and now we have an analytic algorithm making a calculation of the width of this pattern using either a superluminescent diode. This was published in 2023. More recently, we're progressing to use an LED. And so now we can do slide scanning. We can move from field of view to field of view and maintain focus. So we can do multi-well plate imaging of super resolved microscopy using STORM. So this was our first example where, again, we're looking at the Streptococcus pneumonia bacteria in THP1 cells. And these are the super resolved images. The top row here are zooms in of the wide field image and the super resolved image. And in a collaboration with Chris Bakel's group at the Institute of Cancer Research, he was interested in focal adhesions, and he labeled F-actin and Paxilin with ifluor 647 and Alexa 488, and on the top you can see the diffraction limited fluorescence images, and on the bottom you can see the super resolved images. And so here you're progressing towards super resolved high content analysis, which we think can be an interesting prospect for the future. And I won't have time to talk about it, but we're also working on automated cluster analysis to use size of clusters, for example, of chromatin as a readout. And so when you're building super resolved microscopes, it's important to know how well you're doing. And there's this really elegant model that was published by the group at EMBL. And essentially, they are looking at the nuclear pore protein. And this shows the pattern of this nuclear pore protein. You have these two rings separated by 50 nanometers, and in this layered structure, you know that your adjacent clusters of, I think this is NUP96 protein, are separated by known distances. And so what many people do to characterize their microscope is they get this cell line, which has been provided by EMBL, and you label it with your favorite fluorophore, in our case normally iFluor647, and this is showing us that we are indeed achieving super resolution. This is easySTORM implemented with a regular scientific CMOS camera, and we thought, can we make that cheaper? And we noticed that other groups, including Marcus Sauer, had done this, and so we emulated that. And we're working this time with a camera that was originally developed for the amateur astronomy community. So this comes, now you can buy this from Cairn Research Limited, from a company called Altair Astro. This is called the Cellcam Centro. It's a machine vision camera which is fan cooled, costs less than £1,000, and you can see again we can resolve the nuclear pore proteins. And if you want to spend slightly more money, you can get a TE cooled version of this camera for about £3,000, and again you can resolve the nuclear pores. So now we've greatly reduced the cost of the microscope by reducing the cost of the excitation lasers, by reducing the cost of the camera. What's next? Well, you can also, by the way, use very low-cost microscope objectives from companies like Amscope or Polyoptics. You can buy a high-NA 100x oil immersion objective for of the order of £100 or so. And so after that, the most expensive part was the microscope frame. And so we thought, well, why are microscope frames so expensive? What do they need to be? And thinking about this, we came up with this layered structure, which I think of as kind of a wedding cake structure, where basically we have the excitation layer of a fluorescent microscope, which contains the fluorescence dichroic. We have a steering emitter and a tube lens that goes to the camera. If we're having an autofocus, we need a beam splitter to bring in the infrared autofocus beam. And we have a microscope stage and some way of focusing it. And so we built the first prototype, and then we worked with Cairn, with Jeremy Graham and colleagues at Cairn Research Limited, and particularly with Frederick Gerlitz and Sunil Kumar. In partnership with them, we designed this open frame concept, of which the CAD is freely shared. This is an actual microscope, and we can configure a reasonably powerful epifluorescence microscope for between 10 and 15K, depending on how sophisticated you want it to be. And for another 15,000, you can add a motorized XY stage and autofocus for high content analysis. The CAD files of the basic open frame components are shared openly and permissively licensed, and you can buy them from Cairn and now from custom scopes at reasonably low cost. Wherever possible, we use open source software that's already available, ImageJ, Micromanager, and so on. If where necessary, we write our own plugins. And we are hoping that this will be used by everyone, academia, industry, low resource settings where the low upfront cost and the low cost of maintaining the microscopes is important. We think this could also be a teaching resource. And again, you can see details of this on our website. To show that on this open frame, we can still do super resolved microscopy. Again, we image the nuclear pores, and you can see at the bottom left, we again are certainly resolving the nuclear pores. And we also repeated the experiment measuring the width of the glomerular basement membrane for renal pathology. And in this case, we resolved a membrane thickness of below 90 nanometers. And so the open frame microscope, I believe, could be the workhorse of an open source pathology lab, and that would be a vision I would very much like to see realized. So what does the microscope look like? This is a slightly more elegant picture. We now use a manual multi-objective turret, an XYZ stage. Here is our excitation layer. Typically, we might have a camera switching layer where we can slide between brightfield imaging or fluorescence imaging, and this system with a motorized Z stage now would cost about 15K. And this formed the basis of a project sponsored by the Chan Zuckerberg Initiative with the University of Cape Town. We called OpenScopes Africa. And we are basically sharing the know-how on this. This is my colleague Sunil Kumar showing you how this layered structure fits together. It doesn't take just 30 seconds to assemble the microscope. This is speeded up. Ken recently had a competition at a microscope conference where people raced to assemble this microscope to win a prize. Sunil only came third, but I think the winner did it in under 5 minutes. And so as you move from the basic microscope to something more sophisticated, you're building up to a kind of high content platform with motorized XY stage, motorized Z stage. This is our home built laser engine, which again costs about 5K with those laser tack lasers. This is the CellCam camera, micromanager, multiwell plates, and so on. So where does this fit in with other microscopes that are available? Obviously, microscopy in imaging facilities is dominated by multifunction imaging core scopes, and OpenFrame is not aiming to replace these. These are serviced instruments that are maintained. They do the same thing every time you use them. Biologists can go there and use them, and they are there. Unfortunately, for many labs, they're not affordable. There are many specialized microscopes ranging from at the top from lattice light sheet and Minstead microscopes, and at the lower cost end 3D printed point of care scopes, microscopes such as the OpenFlexure project and so on. And OpenScopes isn't really competing with them. Also, if you know what you're doing, you can just go to the Thor Labs or Edmund Optics or other catalog and build your own. And of course, many people can do that, but it takes quite a lot of knowledge to build a microscope from scratch. The OpenFrame microscope fits in the middle, where you can buy all the components. It's designed to directly mount to Thor Labs cage and to be compatible with components from pretty much anyone. But it's a bit like when you buy Lego and you buy back, the garage. Basically you get the bits to build a garage. With OpenFrame, you get the bits to put together to build a microscope. And so you can't build much else, but you can build microscopes. And it works pretty well. It's pretty heavy, so it's pretty stable, comparable to a commercial microscope frame. And so who is OpenFrame for? Well, first of all, it's for me. In my lab, I don't think we will ever buy a commercial frame again, because we typically are implementing our own modalities. And when we do that, we're fighting against design decisions that were made for very good reasons for other purposes. We sometimes don't have full information about the instrument, and of course we would maybe have to overcome proprietary software. And so for myself and for other instrument developers, the OpenFrame is a really good place to start. We're very interested in disseminating this capability to users in lower resource settings. We know that in industry, this is attractive. If you go to microscope conferences, you'll often see open frames in the trade exhibition, because if you're not selling a microscope per se, but some kind of module that fits on it, the open frame is compact and low cost and easy to transport. And there are more than one company selling instruments with an open frame at the core. I think this could be interesting in imaging facilities, but I think we understand that biologists probably wouldn't choose to use this over an existing kind of fully serviced offering, but I think this is interesting to explore. I think it could be very interesting for teaching in universities, and as I've mentioned before, I would really like to see this in hospitals. And so I'm going to talk about some higher end applications. So this is a project with Lucy Collinson's group at the Francis Crick Institute, and they are basically been working on every form of electron microscopy, including volumetric microscopy with ultra microtomes. And essentially, they were preparing tissue labeled with fluorescence in resin, ready for electron microscopy. And they noticed that when they treated samples in this way, the GFP or the M cherry would spontaneously blink, which is interesting because GFP doesn't actually blink that well in standard storm buffers. And so, particularly talking with Martin Jones, we came up with the concept of what if you took some tissue, you embed it in resin, tissue labeled with GFP or some other fluorescent protein, you embed it in resin, you cut it with an ultramicrotome into very thin sections, say 50 or 100 nanometers, and then you excite it, it blinks, and you do storm, so you get better than 50 nanometers XY resolution. So now you've got sub 50 nanometer XYZ resolution of a stack. And so you can do volumetric imaging, and you can also do electron microscopy, so you can do volumetric CLEM. So now this is super resolved CLEM, and your optical image, because it's super resolved, can be registered much better with the electron microscopy image than trying to do that with diffraction limited optical images. And so this works. This is an example of cortical neurons where the mitochondria are labeled with mito-GFP, and the top is showing you the fly-through of the overlaid stacks, and the bottom is a 3D rendering. You could forget about electron microscopy and just use this for volumetric imaging. Then we would call it TomoSTORM, volumetric easySTORM in resin. And this technique, Lucy's vision was to make it usable outside electron microscopy facilities and to lower resource settings. The total system cost, including a benchtop SEM, is only 200K. The microscope costs just over 35K. And one of the challenges when you're doing this kind of volumetric imaging is that if you think about imaging a cubic millimeter of tissue cut into 100 nanometer sections, if you image on one microscope 24/7 doing STORM, it might take you a year. How could you accelerate the imaging? Well, one way you could do it is just to have more microscopes imaging different subsets of the slices. And if your microscope only costs 35K, for the cost of a commercial super resolving microscope, you can have at least 10 of them, so you could go 10 times faster. And when you're thinking about scaling an acquisition time of months to 10 times less, you go from months to weeks or weeks to days. This starts to make these kind of projects much more tractable. And this instrument is now been replicated and installed in Stellenbosch University in South Africa, and we're waiting to see the first results out of there. And so another project I'm going to briefly mention is our CRUK Accelerator project, which had the snappy title, Accelerating Our Ability to Understand and Target Complexity and Heterogeneity in Cancer through Automated Imaging of 3D Cancer Models, including Patient-Derived Organoids, which has the shorter title of MAC3 Cancer. And in this project, we were basically realizing that 3D models in cancer are becoming increasingly of interest for fundamental discovery and drug discovery. But typically, the model is more sophisticated than the readout. Very often, people were using wide field 2D microscopes, just doing live dead assays of 3D spheroids or organoids. And so we wanted to know, could we do 3D resolved high content analysis? And for this, we built 3 instruments: a spinning disk fluorescence lifetime instrument, which is the same as the one published in as our open source instrument. My colleague Chris Dunsby is pioneering oblique plane microscopy high content analysis, and we also decided to build a multiphoton FLIM system, where we call multiphoton multi-beam multi-well plate FLIM. And so the goal here was to explore whether we could find the sweet spot of higher complexity of our biological model with higher throughput for discovery. And so we've come up with many outputs of this project, which I'm not going to have time to talk about, but I do want to show you this. This is a diagram of a kind of open frame on steroids, as well as the excitation layer, the normal excitation layer, the autofocus layer. It's multiphoton microscopy, so we have a layer for non-D-scan detection. This is a layer we are saving for some modality we haven't done yet. At the bottom, we have spectral ratiometric imaging and also quantitative phase imaging I'll talk about later. And the multi-beam aspect, here we have a multiphoton laser, excitation laser, which we use a spatial light modulator to divide into 6 beams. We scan 6 spots on the sample, descan and go to 6 photomultipliers and do 6-channel parallelized TCSPC. And although we have 25% extra loss in the detection path, in principle, this should let us go something like 3 to 4 times faster. And so this works, and I will show you one set of data from when we first demonstrated it. This is imaging spheroids in mouse cells where nuclear histones have been labeled with IRFP and imaged. And here we have a section of mouse lung tissue labeled with the actin and the nuclei labeled with Alexa 633 and CFP. And so you can see that we can do 3D fluorescence lifetime imaging. I'll say no more about that, but it's an example of the open frame not just being used for lower cost instruments, but being used for very powerful instruments that you probably couldn't implement on a commercial frame. So getting back to dissemination, and which is the main drive of our open source work, we were fortunate to have this CZI funded project with colleagues Digby Warner, Karen Jacobs, and Michael Reike in the University of Cape Town. And we basically were exploring rolling out open source microscopy across Africa. And to this end, we first of all hosted an online workshop where we had 155 attendees, mostly from Africa. And we also had an in-person microscope workshop in the University of Cape Town, where we had 22 delegates from 11 African countries. These are they. And the experiment, one of the activities that we undertook in the workshop was we got them to do acquire two color Z stacks of fluorescently labeled samples using Micromanager, and then display the images in ImageJ. And then we made them strip the microscope down to its component pieces and then put it back together again, and again acquire Z stacks, two color fluorescent Z stacks, using Micromanager displaying in ImageJ. And they did this all inside a 45-minute session, and we got a lot of very positive feedback from this. These colleagues typically would never have been allowed to strip down the microscopes back in their imaging facilities back home, and indeed many of them said they would never have the confidence to attempt such a thing. And so this really fires us up with enthusiasm to expand OpenScopes and the OpenFrame. And of course, it's not that easy. There are many challenges in low middle income countries, including in Africa. As well as the instrument, you've got to find funding for the import costs, the reagents. If you're doing something like STORM, you need to make sure you've got the safety infrastructure in place. You've got to get your local administration to buy in, to give you that funding and the time to do it. You need to incentivize bright people to come and work on this. Ideally, we don't want this to be funded by donors. We want this to be funded by regional funding, and we want to kind of bottom up. We want to stimulate a bottom-up demand for these techniques. But this is limited because many people don't have that much experience and therefore knowledge of the potential of microscopy. And many of the people who came to our microscope had not even ever worked with a digital microscope. So to address this image, we came up with the Open iCam. This is very simple, particularly developed by Sunil Kumar. This is a 3D printed kind of gadget to mount a cheap camera onto the eyepiece of any microscope, and then a USB cable to a laptop where you can use Micromanager to acquire images and ImageJ to display them. So this essentially, for £200, lets you convert any scope to digital microscopy. And we sent one of these kits to everyone who had attended the workshop. And if you want to know more about this, you can see it on our OpenScopes webpage. And so one of the people who came to our workshop was Daniel from Meru University of Science and Technology, and subsequently in a British Council funded project, he came to Imperial to learn about the OpenFrame microscope, and we shipped it back to Kenya. And this is Daniel and colleagues in Kenya, where they're using this microscope to in various applications, including malaria and cancer research. And so this is the basic OpenFrame microscope, and this is Sunil building it again. And our colleagues now at the University of Cape Town have come up with this OpenScopes traveloscope thing, where they have built a kit which fits into flight cases, which they are now rolling out to multiple sites in Africa, which they intend to be local nodes for clusters of microscopy. This project was funded by the Wellcome Trust, and we're currently seeking funding to renew that so we can widen that out to more people. So this basic OpenFrame microscope, which now looks like this, our colleagues again in Cape Town are using this for their own research in tuberculosis, and these are some recent images. This can be upgraded to a more sophisticated microscope, which we are now working to install in containment level 3 facilities for tuberculosis research, both in the University of Cape Town and also at Imperial, for doing long time-based assays. So again, it's multicolor imaging, autofocus, cell can kicker, and also quantitative phase imaging. Tobias was interested in microscopes. This is a picture of that M3M microscope at the CRIC, the multi-beam multiphoton microscope. And here we built an incubator, which is basically a sandwich between two optical breadboards. So we can build anything we want, any kind of transillumination, light source detector. We can build anything we want inside here. We've actually got a optomechanical device to feed multi-well plates out so they can go to a robot arm into a high content system. And so we think this is a very flexible approach, because often when you're trying to modify commercial microscopes, modifying the microscope incubator can be a major challenge. But for the open frame, this is probably too expensive. And so we have, particularly for the tuberculosis and research, we come up with two incubators that we 3D print. We use simple foil heaters. The top left one is one that we have used for a microscope slide, a manual microscope slide microscope, and the bottom one is one that Sunil is currently working on to work on 96 well plates, which will be used in the time-lapse microscope that we're going to install in BSL3. And so I've mentioned quantitative phase several times, and I'll briefly go through our technique for this. We're using the technique of differential phase contrast microscopy, and this is a technique that is not new. Colin Shepherd, who else came up with this in 2009 with Shalin Mehta, and basically the concept is that you have a standard transillumination microscope setup, and you either put a mask in the back aperture of the condenser, or you can put the light source and have a segmented light source. And Tian and Waller did this very elegantly with an LED array. And so essentially what you do is you block half of the mask and take a picture, block the other half of the mask and take the picture, do this in both directions. You acquire 4 images of the sample illuminated with oblique illumination, and you then calculate the difference images, and using a weak object approximation deconvolution you can calculate the phase transfer function, and hence you can generate quantitative phase images. And so this is how it works. You put the mask in 4 times, you generate these 4 images, and you generate these quantitative phase images. So we were interested in this, but we wanted to make it single shot. And we had just become apprised of the PolarSense camera. So this is a camera now available for as little as $1,000, which has a mosaic mask where instead of a Bayer filter giving it color imaging, it gives polarization resolved imaging. It has polarization filters organized at 0, 90, plus 45 and minus 45 degrees. With respect to the edge of the frame, and so it acquires 4 interleaf polarization resolved images at once. And so we inserted a polarization mask with 4 quadrants where we have a polarization filter parallel 40 plus 45 minus 45 and 90 degrees, and we make these polarizers out of cheap polymer film which you can buy for a few pounds. And basically this works, and essentially in a single shot we acquire these 4 interleaved images from which we can either synthesize the images that we would have got if we'd done it in the conventional way, or following the work of Hui Liu in our group, we can use a matrix formalism to calculate the reconstructed phase. And you can see more about this in our published papers. So this gives us single shot quantitative phase imaging. This is an example. Again, this is a surface plot. This is a line scan through it. Because it's single shot, we can run as fast as the camera frame rate. Which if you buy the more expensive one of these Sony camera sensors, it's something like 70 frames a second. This is applying it to live C. elegans, and you can see it's digesting something here in this particular organ. Because it's using it's not wavelength specific, you can use it in any spectral window. Typically we use the red, so this is an overlay with a GFP labeled C. elegans. And to show the dynamics, this is an image of water droplets dropping on a cover slip, evaporating, so you can see the time scale. This is imaging quite quickly. You can of course implement this on the open frame, and we now have what we call OpenTX. We're making a modular transillumination light module, and one of the modules is just to put our quadrant polarization mask in here. And here we have a microscope where we illuminate with blue excitation light for fluorescence and red excitation for phase, and that was how we acquired that dual color image in the previous slide. We can also do time-lapse imaging. This is a movie speeded up of 8 hours time-lapse of live kidney cells. And this is looking at M. smegmatis. And these are just 2 frames from a very boring 4-hour movie where not much happens, but there's a little bit of growth in this region here. So in this technique, and any version of diffuse phase contrast microscopy, one of the constraints is that the numerical aperture of the illumination has to be greater than that of the imaging with the objective lens. And so We've also come up with a higher NA version of this, where we do away with the condenser and we synthesize an extended light source using a standard domestic light bulb costing £7 and a low-cost color filter and our quadrant mask. And here we get up to 0.8 NA, and so this is 60x quantitative phase imaging. And this whole setup costs £200 plus the cost of the polarization resolving camera. And people have asked us how quantitative this is. So this is using the standard lower cost benchmark technologies chart. So this is the one that's printed from the photolithographically made mask. And this is showing how the gray is the expected phase profile, and the red and green lines are the calculated phase profile without a correction applied. And you can see that up to half a radian, it's pretty quantitative. Above that, it's at least semi-quantitative. And so this is a technology that we can use widely, and it doesn't have to be implemented in an open frame microscope. So I'm now going to tell you about an example where we're using it at the Crick, and this is building on work with Eric Sahai's lab, where Alex Lemarrois has been working on single cell resolved time-lapse microscopy, particularly using FRET to read out activation of the ERK pathway to look at responses to cancer drugs. And so basically, when you apply a cancer drug, say an ERK inhibitor, most of the cells are affected, but there will be a few that don't respond. These are the outliers that maybe come back as resistant cells and relapse, bring you back to the disease. And the top is an example where these blue cells are the rogue cells. Here you have all the cells responding with a certain FRET response, but these are the outliers. And what Alex did in this experiment was she segmented the cells, every single cell over time, and these were over many even tens of hours, multiple days, and she looked at how the FRET signaling varied over all this time for every cell. And basically, she discovered that the pulsatile behavior, the switching on and off of the ERK pathway, was a phenotype, was a characteristic of the cells. She then did an siRNA screen, knocking out different genes to see which knockout would have the biggest effect on this pulsatile behavior. And in a paper which is currently on bioRxiv and under review, she associated the genes associated with the endocytic machinery with the recovery of the ERK activation. And so this is not what I understand. This is cancer biology and not physics, but this is an example of long-term time-lapse microscopy. But the challenge in doing, and this was done on one of our microscopes, not in this case an open frame microscope, using time-lapse FRET. But the challenge here is that if you're imaging over many tens of hours, you really have to think about phototoxicity, because fluorescence imaging is phototoxic. The more times a molecule goes through the fluorescent cycle, the more chances it has to get trapped in a triplet state and generate singlet oxygen. And so we thought, can we use label-free phase imaging to segment and track the cells instead of using fluorescence? And so we can. And in our lab, we have an Olympus IX83 microscope, and we made one of our polarization quadrant marks and simply inserted it into the already existing filter holder in the transillumination pillar. And off we go. We can do quantitative phase imaging. Our student, Karishma Valan, did a comparison of phase segmented imaging against fluorescent segmented imaging using CellPose, which is an open source program which is exploits machine learning, and using the TrackMate program she could then do segmentation. And what she found was she could track not only the ERK activation of every cell over time, but also from the quantitative phase image she could calculate the dry mass. And it turns out when you're looking at the response of cells to cancer drugs, you can not only classify different responses with respect to the ERK levels read out by FRET, but you can also get some kind of signal from dry mass. And so integrating quantitative phase with fluorescence functional readouts seems to be interesting. Another application we thought of was resolving cell cycle. And so in this experiment, we implemented pDPC again on the microscope, and we imaged a cell that was expressing a fluorescent cell cycle marker. So this was PCNA, which is a fluorescent cell cycle marker where the fluorescence M-scarlet fluorescent protein undergoes translocation from between the nucleus and the cytoplasm during different phases of the cell cycle. And so we can image cells over multiple cell cycles using both fluorescence and quantitative phase. We can use the fluorescence as the ground truth to train a machine learning model, and then use the quantitative phase to read out. And this is our kind of a matrix of confusion where we can get approximately 90% accuracy using machine learning applied to a quantitative phase image to classify the cells in cell cycle. And so I think this could be potentially very powerful, because it means whenever you do a fluorescence experiment, you can also do quantitative phase imaging, and you don't have to synchronize your cells. You can retrospectively align all of your fluorescent readouts to cell cycle. And then the last topic I'm going to talk about in this talk is going to be our single shot hyperspectral imaging technique. So this is a technique we call PulseSpec, which you'll see for obvious reasons. So hyperspectral imaging, to remind you, is where instead of just imaging a fluorophores or other sample brightfield images in different color channels, you image in many closely spaced color channels such that you acquire a spectrum in every channel, or if you're doing hyperspectral imaging, you have a spectral in every pixel. So this is interesting, but obviously collecting all this information can take a long time. And so there's been a lot of interest in using spectral phasers. People like Hans Gerritsen's lab and Enrico Greton's lab have really explored this, and others. And basically what you do is you kind of do a Fourier transform where you multiply your spectrum by a sinusoidal or cone sinusoidal function, and you plot the outcomes of that on a plot. And this is you would call a phasor plot or a phasor vector plot. And basically you have your XY image telling you where your fluorophores are, and on the phasor plot, every pixel maps to a place on the phasor, which depends on the width and the spectral the mean wavelength and the width of the spectrum. And so this is a way of doing data reduction, and it begs the question, if this is how I'm going to analyze my data, why did I collect all that data in the first place? And Enrico Greton's group showed that you don't have to. You can actually take your fluorescence image and you can modulate it with either a cosinusoidal function or a sinusoidal function in spectrum, so transmission versus wavelength or transmission versus frequency, and you can measure the power transmitted, and from that you can directly construct phasors. And so this is of course much more photon efficient. It will work wide field. It's not messed up by scattering. This has also been extended to a single shot technique by Wang et al. And so this is spectral phasors. And we looked at this and thought, that's very interesting. It doesn't have to be sine or cosine. It can probably be a range of different functions, P and Q, as long as they're orthogonal. And it would be great if, as was the case in the paper by Wang, you collect both the signal transmitted through your modulator and the signal not transmitted or reflected. So you collect IP and I1 minus P, IQ and M1 minus Q. And then you can construct what we would call a spectral modulation vector, of which a phasor is a special case, and you can do spectral modulation vector analysis. And so why do we do this? We do this because we wanted to replace color filters or dielectric coatings with polarization optics, with a Lyot filter, where we basically have a retarder between two cross polarizers and or two parallel polarizers, and basically the transmission function of this Lyot filter is cosinusoidal, and you can configure it more easily to do. It turns out to be tricky to do sine and cos. You need an achromatic quarter wave plate, but you can do cosine and cosine theta and cosine 2 theta, and these are still orthogonal functions. And so using a retarder of lambda naught and of 2 lambda naught, retarders at 45 degrees, we can measure the output from polarizing beam splitters, and we get our 4 parameters to plot our spectral modulation vector. And in this sense, this would be a single shot lossless technique using polarizing beam splitters. This is a little bit cumbersome to implement, and we remembered the polarization resolving camera. By the way, I should say that this technique has also been independently thought of by the group of Stegman et al. And published in a similar time. We decided to implement this with the polarization resolving camera, and here you can implement it simply with a low-cost polarization film, a retarder at 45 degrees, another retarder at 0 degrees, and this gives us these spectral modulation functions, which turn into essentially cos theta and cos 2 theta. And these retarders are also made from low-cost polymer film, and so this can be just basically mounted on the front of this polarization resolving camera. And does it work? Well, we showed that it worked by inputting the output of a tunable light source, a supercontinuum, and this is not playing. I don't understand why. And so this shows you how the spectral modulation vector is mapped out as we tune the wavelength of the supercontinuum. And we tested it with a color test chart generated on a mobile phone display. This is the intensity image. This is the spectral modulation image, where you can see the different clusters of points corresponding to the different colors. And then you can reconstruct the color image from the spectral modulation vector. So we thought this would be interesting to explore for some application like pathology. And so we took an H&E stained histopathology slide. We imaged it with a regular red, green, blue camera, and we get the column on the left-hand side. We looked at the total intensity signal, which is the second column. We imaged it with the polarization resolving camera-based pulse spec technique, and now you see the spectral modulation vector plot. You see the different clusters corresponding to different quantities in the H&E image. This is the image that we generate directly from the spectral modulation vector. And if we do a white color balance, we can reconstruct the image that we would have got from the RGB camera. So you might say, well, what's the point if you've already got an RGB camera? But this can work in any spectral region. We don't have to use the wavelength bins provided by the standard biofilter. We could use it in shortwave infrared, for example, as the group Stegman did. We can use it with exotic CMOS cameras or any other camera that we want to use. And we believe that this is a flexible way to do multispectral, or if you like, hyperspectral imaging with vectors. And so just to wrap up this talk, I'm going to summarize why would you want to use modular, low-cost, locally sustainable microscopes? Well, first of all, they're cost-effective. Open source hardware, permissibly shared CAD files can be used to make basic components. The other components you need can be low-cost and generic. We often buy things from Amazon or Alibaba, but you can also buy things from your main optics suppliers. You only need to pay for the functionality you need. You can reuse what you have when you don't want to use it. So our sales motto, although we don't actually sell anything, might be less for less. If you completely reproduced the functionality of a commercial multi-user microscope, it would probably cost you more doing it with an open frame. But if you just want to do STORM or you just want to do quantitative phase, you can buy the bits for what you need to do, and it will almost certainly cost you less, and you'll be able to maintain it. If you build it, you can maintain it. However, like open source software isn't free software. Someone once pointed out to me, you have to pay someone who's able to install it and get it working. Open hardware is not free hardware. It does need users to do more than they would need to do for commercial service microscopes. For many users, it's attractive for rapid implementation, but it does require some understanding of microscopy. It's sustainable. If you can assemble the microscope, you can maintain it. This means you don't need service engineers, you don't need service contracts. And this is, of course, really important for those countries in the world where service engineers either don't go or where it's prohibitively expensive to get them to come. And critically, there is minimal obsolescence. Once you've got the microscope and you want to upgrade it, you don't need to buy a new microscope. You just pay for the components you need for the new functionality. And I would expect our microscopes to last decades. It's flexible and extendable. We believe it gives research grade performance. You can image almost any microscope modality. We have implemented quantitative phase, FLIM, multiphoton, high contact analysis, slide scanning, hyperspectral imaging. You can buy commercial components, including all the core elements, or you can make your own, and it's open to all. No more scopes to landfill. How do you need to use it? Well, I would say that if you're a biologist, you need to at least have an awareness of building microscopes or partner with physicists, engineers, computer scientists who are able to do the microscope assembly, operation, image acquisition, and data analysis. It's not the same as using a regular bioimaging core, where a biologist gets a full service. This is probably better while you've got it and while it's supported. But I believe that this is good enough to get a lot of science done sustainably and at scale. Why is open microscopy exploding now? Well, I think it's obviously because new technologies have made previously expensive instruments much more accessible. Computers, of course, are almost free if you've got a mobile phone. We can get powerful GPUs in gaming computers, which we can use for rapid reconstruction. FPGAs have great promise for the future. We can access all sorts of open source software and resources on the cloud and the internet. Detectors are getting cheaper and cheaper, particularly cameras driven by advances in mobile phones. Light sources are getting cheaper. LEDs are available at almost any wavelength now, leveraging consumer electronics. We can piggyback optical telecoms for fiber optics and other components. As I've mentioned, open source software. I think we still have a plethora of open source software, but you can, even if you just stick to Micromanager and ImageJ, you can do a lot. And then there's Python and Apari and etc. You can use open source CAD resources. And so you can make these different modalities, which could be important for lower resource settings, which might include lower income countries. But in the UK, I would describe the National Health Service as often a lower resource setting. And in my dreams, we would be able to address some of their problems using these technologies. So I'd like to thank you for your attention. I'd like to acknowledge the many people who have contributed to this work and answer any questions.